Welcome everybody to the fourth contributed talk session of Bioconductor. We have four wonderful speakers giving 10-minute talks today, followed by a roughly 15-minute Q&A session. Please submit your questions via the Pathable poll feature and make sure you note which speaker you're addressing your question to. Our first speaker today is Anthony Mammoliti. And I will start that video now.
Hi, my name is Anthony Mammoliti, and I'm a master's student in Dr. Benjamin Habe Kane's lab at Princess Margaret Cancer Center. And today, alongside with Minoru, who is a software developer in the lab, I will be discussing our platform called ORCESTRA, which aims to achieve research reproducibility in pharmacogenomics. First off, I would like to acknowledge that there are many current challenges today in the field. There are large amounts of data that is being generated, and therefore the analysis pipelines and interpretation of this data is becoming increasingly complex. In addition, there are a plethora of new computational pipelines that are being released to process this influx of data, which is often very difficult to keep up with. Therefore, it is being said that we are facing a reproducibility crisis. There are many notable pharmacogenomic data sets over the past few years that have been released. And this includes GCSI from Genetech, CTRPV2 and CCLE from the Broad Institute, Gray from the Joe W. Gray Lab in Oregon, FIM from the Institute for Molecular Medicine in Finland, UHN Breast from the University Health Network in Toronto, and GDSC 1 and 2 from the Sanger Institute. Essentially, our goal is to utilize these panel of cancer cell lines from the large pharmacogenomic data sets, which include drug sensitivity screening, and molecular profiling for biomarker discovery. Therefore, the main challenge is how can we process and share this complex data using a plethora of pipelines in a way that will achieve reproducibility and data provenance. To solve this issue, we developed a platform called ORCESTRA, which is a cloud-based platform that allows for the transparent, reproducible, and flexible analysis and sharing of pharmacogenomics data. It integrates both genomic and pharmacological profiles of cancer samples through automated and versioned processing pipelines using a tool called Pachyderm. And at the end, it creates a customized and fully documented data object known as a pharmacoset or pset for future analyses using the PharmacoGX bioconductor package. As a reference, a pharmacoset, also known as a pset, is a multi-layered data object which includes cell and drug curations, drug sensitivity, and molecular profile data, which all can be used for biomarker discovery and drug repurposing. At the heart of ORCESTRA lies Pachyderm, which is an open source tool that allows for the deployment of PharmacoGX pipelines to produce these psets by storing data in repositories. It utilizes Kubernetes and a cloud or on-premise cluster environment. Versions of a dataset and pset can easily be tracked through unique identifiers that are assigned to them by the tool. If there's ever change in a repository, such as through the modification and/or addition of a file, this will automatically trigger pipeline deployment, and therefore we will get a new version of a pset. Ultimately, ORCESTRA hosts 7 pharmacogenomic datasets, which include CCLE, GCSI, GDSC-V1 and V2, CTRPV2, Gray, FIM, and UHN Breast, all with customizable molecular and pharmacological data on the platform. And this gives rise to the exploration of 1,676 unique cell lines, 928 unique drug compounds, and 1.5 million drug sensitivity experiments. We process all of our molecular data on our on-prem HPC using SnakeMake. This data gets piped into our Microsoft Cloud infrastructure where Pachyderm exists and versions all of our data and pipelines. Every file that exists in one of the data repositories within our Pachyderm cluster is versioned with a unique ID, and this is represented by the red circle in the diagram. The data that is hosted within our Pachyderm cluster are the process molecular data, cell line annotations, drug annotations, raw drug response data, and processed drug response data. Users can customize the content of their pharmacoset, which includes molecular data that is processed with different genomic tools, such as Callisto and Salmon for RNA-seq data, and different versions of drug response data for a given data set. However, for each user selection, Pachyderm will combine the various version pipelines together to form a versioned pset. However, just say that there is an update to the molecular data for a given data set. We will then push this data automatically into our Pachyderm cluster, and this will result in a new unique ID for only the molecular data, and therefore a new unique ID and versioned pharmacoset. What is very interesting about Pachyderm is that it will not reprocess any of the other pipelines that are required to build a pharmacoset, which comes in handy as we don't always want to reprocess pipelines that have a heavy computation time, such as the drug response data. At the end, this pharmacoset that is now versioned will automatically get pushed to Zenodo, which is an online data sharing repository where it is assigned DOI. At the end, the custom pset with its associated DOI can be accessed on ORCESTRA with a custom metadata page that is sent to the user via email. Now Minoru will show you a demo of the ORCESTRA platform.
Hi, my name is Minoru Nakano, and I would like to go over a brief demonstration of the ORCESTRA platform. Welcome to ORCESTRA, a cloud-based platform that provides a transparent, reproducible, and flexible computational framework for processing and sharing pharmacogenomic datasets, or psets. You can find the list of the latest version of psets that have been created by our lab in the List of Canonical Psets page. You can view detailed information about each pset by simply clicking on the name. Single pset page, such as this one for CCLE 2015, provides metadata and molecular data sources used to create this pset. You can also view information about the pipeline used to create this pset under the Pipeline tab. You can download the pset by simply clicking the download button. You can click on the DOI to go to this pset's Zenodo page. If you would like to search existing psets or request a customized pset, go to Search and Request page. You can search through the existing psets by filtering them with the pset parameters panel to the left. If a pset with a desired set of parameters does not exist, you can request ORCESTRA to generate it by switching the Request pset toggle. You can submit the request after filling out additional parameters, pset name, and an email address to receive a notification for the pipeline completion. Your request will be stored in the database and will either be processed immediately if the Pachyderm data processing layer is online, or will be processed when the layer becomes available. Once a pipeline request is complete, you will receive an email which includes a link to the pset page in ORCESTRA, a link to the Zenodo page, and the link to download your pset. ORCESTRA has additional features such as dataset metrics and pset usage statistics, and API endpoints to access information about existing psets. To explore the platform, please visit www.orchestra.ca.
Thank you for your time. I would like to thank Dr. Benjamin Hapekins and my lab members for their immense amount of support and guidance throughout this project.
Wonderful. That was a great way to start us off. Our next talk is from Shraddha, who will be giving her talk live.
Well, that was a classic Zoom moment. Thank you, everyone. This is very exciting to be presenting at Bioconductor this year. My name is Shraddha Pai, and I work in Gary Bader's lab at the University of Toronto. Today, I'd like to share with you our software, netDx, which helps you build an interpretable patient classifier by integrating multi-omic data by using a patient similarity network. I will walk you through these concepts. So the context of this work is that netDx aims to provide software to help clinical researchers and basic researchers create risk models. Risk models can be used to predict drug response, prognostics, and risk, disease risk, and so forth. And a key part of risk models is the integration of different types of data, multi-omic, but also other modalities like clinical data, imaging data, and so forth. So what netDx does is, when provided with a set of different assays and or different data types, netDx uses machine learning to allow you to predict patient outcomes. You can have binary outcomes or N-way outcomes. At present, we support categorical outcomes only. You can use machine learning to classify new patients with accuracy, identify which of the data you provided for the predictor was actually informative in making the prediction, and help you generalize to other types of data. How do we achieve this? So this is where the patient similarity networks come into play. A patient similarity network is simply a network where the nodes are patients and the edge weights quantify how similar the patients are for a given data type. So in this example, we've got 3 cases and 3 controls: clinical, genomic, metabolomic data. The patient similarity network for clinical data shows that the cases and the controls separately cluster very well, but for metabolomic data, only the cases cluster very well. So what is similarity, I'm often asked? Similarity could be as simple as Pearson correlation of gene expression measures between 2 individuals, or it could be other measures, whatever makes sense for your application. So what netDx then does is, when provided with heterogeneous patient data and some labels of interest or classification, it takes these heterogeneous data and converts them to a common space of patient similarity networks. It then uses a step from machine learning called feature selection, which essentially puts a score on each of these features to tell you how predictive they are. A threshold is then applied, and then they're using the top features, a final integrated patient network is created. A new patient is then classified based on which group they are most similar to in this integrated network. So netDx allows you to identify predictive features and to classify new patients. The methods behind netDx were published last year. I've got a link here, and I will make this talk public after this session. So in the methods paper, we benchmarked netDx using the pan-cancer survival data. So here we've got 4 different cancer types: kidney, ovarian, lung, and brain. The task was, given 5 different omic data types and clinical variables, predict if the patient is going to have good outcome or poor outcome in terms of survival time. And each of these plots shows you the performance for netDx, which is in pink here, as compared to other machine learning methods. And we found that in this benchmark, netDx outperforms most methods most of the times. There are some exceptions, like when the SVM method, support vector machine, finds a really non-linear separator for some instances. Not surprising, because no machine learning method is going to fit all situations, and you can consider netDx one tool in your toolbox for clinical prediction. Importantly, When provided with omic data, netDx provides native support to create features at the level of pathways. So before I showed you that you take, for example, gene expression data and you make a single PSN, patient network, out of it. But what netDx allows you to do is pull pathway definitions from curated databases and break up this gene expression matrix into one feature per pathway. What this does is when you now run feature selection, you are scoring pathways based on their predictive value for clinical outcome. This improves interpretability and is a good starting point for rational drug design. Here's an example that was part of our methods paper. The task is to classify breast tumors as being of luminal A subtype or a different subtype. Luminal A subtype is a type of breast cancer. It's a breast cancer subtype with good prognosis. And so here we start with just mRNA data, and we break it up into 2,000 pathway level features, and we run netDx. The predictor does really well at the end of the day in almost perfectly separating tumors as luminal A or other subtype. But this is what you get at the end of the predictor. It isn't just doing well and you don't know how. This visualization is called an enrichment map. It is showing you which pathways and pathway themes scored well at predicting a tumor subtype. So each of these nodes is a pathway, and it is colored by how well it did in the algorithm. Pathways that share genes have an edge between them, and then you can use clustering approaches to find themes that do well. When we do this with netDx, we find that the pathways predictive of breast tumors are consistent with the known dysregulated cellular processes in this disease. You get terms like DNA damage repair, DNA synthesis, cell cycle related terms. So you can then take this and drill down into specific pathways and ask the question, what is it about this pathway that's making it predictive? It generates hypotheses for downstream experiments. So how netDx works? I told you already that it uses machine learning, and this is what it does. Given the user provided data, netDx breaks the data into training and test partitions. The model is trained on the first partition, and this is a blind set on which it is evaluated. Like I said before, you start out with the data, and it all gets converted into patient similarity networks. We then go through a step of feature selection where networks are scored from 0 out of n, right? Whatever your cutoff, whatever your n is for your application, like 10. For each of these 10 iterations, netDx takes a different sample of training data, makes the PSNs, and then runs a kind of regularized regression, which tells you which of the networks have a non-zero weight and which don't. Networks that have a non-zero weight have their netDx score bumped up by 1. You do this again and again for different samples of training data, giving you a range of scores for features. You then apply a cutoff for your features and say features that score 9 out of 10 are my selected features. Now you can take your training and your test set, just the selected features, and make a single integrated patient similarity network. netDx then runs label propagation on this network, which basically walks from known examples of class A down to all test patients, and known example of class B to all the test patients. And then you ask, well, which class is the patient similar to? And this is how classes are assigned. And the model is evaluated. This whole box gets repeated, this whole process gets repeated 100 times or however many times you'd like in order to find which predictors are, which features are consistently predictive and how well the model does in generalizing to new types of data. So this is how it works. netDx is in BioC as of version 3.11. I encourage you to try it out and give us your feedback. We've got wrapper functions to build predictors, so simple functions that say build predictor, these many splits, here are my features. There are functions for organizing features into pathways. It uses multi-assay experiment object type to take the user provided data. It's a bioconductor object container for different data types. It also plugs into the network visualization software Cytoscape. using the RSci3 package. This allows you to use netDx to visualize integrated patient networks and the network of top features that I showed you. So there are vignettes showing you how to build predictors with multiple data types, sparse genetic data, and so forth. Current disease applications, very briefly, we've used it for predicting antidepressant non-response, prognosis in pediatric traumatic brain injury. And in the future, we're planning to extend it to continuous valued phenotypes and build a deep learning variation for increased tunability. Thank you very much. With that, I'd like to acknowledge my lab, Gary Bader's lab. Lots of people contributed, lots of external collaborators from Munich, Verona, and Quaid Morris at U of T for good discussions. Thank you very much.
Wonderful. Thank you for a very interesting talk. Just a reminder for any of the attendees that came in late, please submit questions using the Pathable poll feature and make sure to note which speaker your question is being addressed to. Our next speaker in the session is Gabriel Odom, who is also doing a live talk. Gabriel, take it away.
Thank you. I'm so happy to see everyone here. I'm going to go ahead and share my screen, and you should be all seeing slides at the moment. So thank you all for joining us. I want to first give comments and commendations to everyone for surviving a global pandemic as well as possible. So my thoughts and my hearts go out to all of you. I hope that next year we're able to do this in person. So what I'd like to talk to you today is using Pathway PCA, which is a bioconductor package, to perform a global test for integrative pathway analysis for multi-omics data. I left the link to this to the slides in the chat on Pathable. I'm part of Lily Wong and Stephen Chen's lab at the University of Miami Sylvester Cancer Center, and I'm also an assistant professor at Florida International University. So I'm going to go over a brief outline of what we're going to talk about, start with the problem, current approaches, and some of their limitations, briefly discuss our method and some short simulation results and a practical example. So the motivating problem, as we've seen before, as we've seen in these two previous really well done talks, genomics data suffers from the curse of dimensionality. And we often want to use matrix decomposition techniques to reduce the dimensionality from tens of thousands, hundreds of thousands, to something that's considerably more manageable. However, statisticians often get into a lot of trouble whenever they use matrix decomposition techniques without regard for existent biological information. Such very commonly used biological information would be pathway information, biological pathways from, for instance, MSigDB or KEGG or WikiPathways. And so we see that pathway analysis is a wonderful technique that will allow us to combine biological information with the data. Pathway PCA is one tool that we can use to bridge the gap between matrix decomposition techniques and biological information. So the Pathway PCA package we were able to publish on Bioconductor last year. At BIOC 2019, last year, we gave a workshop that shows how to use this, this package for your own research. So I encourage you all to check out this link here. And so the current approaches, I have two schools of thought on this. One is a one-platform supervised approach. The other is a two-platform unsupervised approach. So the idea would be that we have proteomics data and we have RNA-seq data that we're trying to relate to a clinical outcome, let's say, for instance, overall survival or microsatellite instability, something like that. And we want to take advantage of prior biological information through pathways. So the one platform supervised approach would be to, for instance, use Pathway PCA or some other pathway analysis technique to find which pathways are related to the biological response. However, the limitation there is it's not, we can't make systems level intuition. We can't have systems level intuition about the survival response because we can't incorporate this, for instance, proteomics data. So it's good for one particular platform, but we can't go across platforms. On the other hand, if we use two platform matrix decomposition techniques such as non-negative matrix factorization or sparse canonical correlates analysis, then we're able to dig into how pathways work together across platforms of data, which allows us to look at the system as a whole, but it's difficult, the weakness there is it's difficult to incorporate clinical information to ascertain or give value to say, okay, well, this cluster of genes and proteins that we found is actually related to the overall survival. So our technique here is, given two platforms of data with matched samples, let's extract using pathways an eigengene and an eigenprotein from each particular pathway, and then we're going to fit some form of linear model or Cox proportional hazards model, depending on the type of outcome that you have, that allows us to integrate information from the RNA-seq data and from the proteomics data. So this is a global test because we're testing against the global null that neither of the relationships are there. So this that beta 1 and beta 2 are both equal to 0. To see a detailed walkthrough of this process, we've published a paper in Proteomics, and we included the vignette to do this process as some of the supplemental material. So this is the proteomics paper that I just mentioned. It was published 2 months ago, so we thank you to the proteomics reviewers. They gave us some great feedback and really helped tighten up our paper. In that paper, we use a simulation study to compare how the Pathway PCA global test is doing against other matrix factorization techniques. So the Pusher et al. Paper from last year compares a couple of different multi-omics techniques, and we took their two best performing methods, which were the non-negative matrix factorization and sparse canonical correlates analysis, and compared power and test size. As we see here, the test sizes were all reasonably well controlled, less than 0.05. The power, what we did was we generated this synthetic data following this example paper, and we chose a subset of pathways to treat. And the design points were to change the proportion of features in the pathway that were treated, and also to change the treatment effect. So in this figure, the columns of these 4 columns of figures are the proportion of treated features in the pathway from 10% up to 50%. And the feature rows are the strength of the treatment from 2 times the standard deviation up to 6 times the standard deviation. The power is on the vertical axis. The horizontal axis are the 3 methods that we compared. So on the left side, in the salmon color, is the global test, which is our method. In the middle, in this green color, is the non-negative matrix factorization. And on the right side, in this blue color, is sparse canonical correlates analysis. So as we can see, moving from the top left down to the bottom right, so as we move diagonally across this grid of figures, as we increase the proportion of features treated and the strength of the treatment, all of the power for these methods increases, which is great. We also, however, see that across the board, the global test method is able to have higher power, outperform this classification exercise than the non-negative matrix factorization and the sparse canonical correlates analysis. And we strongly believe that this is because this technique is allowing us to use the clinical outcome. It integrates the clinical outcome with the 2 platforms, both RNA-seq and in this case, I believe it was DNA methylation. As a practical example, we used some ovarian cancer overall survival from the TCGA repository. We're using the linked omics hosting site to get that data. And we performed independent pathway analysis using wiki pathways, which, if you haven't seen, there's a link here. There's a great R package, bioconductor package, R wiki pathways, that connects to the wiki pathways site. And it also plays well with Cytoscape, which you should really check this out. It's good stuff. But we performed independent pathway analysis, and we found that after false discovery rate adjustment, none of the pathways were dysregulated for this particular cancer. And that's simply because proteomics often, you have very small number of samples. Sometimes it's very difficult to piece out the signal. once we used the global test to perform an integrative pathway analysis, we were able to find 2 pathways and confirm that these pathways made sense biologically, and showed that these pathways were indeed related to overall survival for ovarian cancer. And we show this in section 3.3 of the paper. At least these pathways are very much worth investigating. So to wrap up, I wanted to be sensitive of your time. So we've shown that the Pathway PCA global test is a technique that we can use to integrate 2 different platforms of data with a clinical response. That clinical response can be classification, regression, or survival, and it will use the eigengenes and the eigenproteins, if we're using protein data, these eigenvectors from the pathways for these 2 platforms to relate the pathways to the clinical response. Now, for future work, we'd like to reduce this limitation that we have on the matched samples, and so we're looking to move to 3+ platforms and reducing the limitation necessary that we have these matched samples. So pay attention to that. That we should have some results that will be coming out in the next few months. We'll be building this current functionality and the next functionality into Pathway PCA version 2. If you'd like to see what we're working on now and what we've worked on in the future, this is my PI's lab, transbioinfolab.org. And I'll leave you with a couple of closing remarks. First of all, I have a lot of people to thank, too many people to really fit on the slide. But my co-authors, they've been fantastic. The reviewers at Proteomics, also fantastic, very helpful. And if you'd like to connect with me, I'm rev.gabriel on Twitter. If you'd like to see the work that we're doing related to the coronavirus response in South Florida, we've got a website called the miamicovidproject.com. So stay safe, wash your hands, wear a mask, all of that stuff, be healthy, and I hope to see you all in person next year.
Great, thank you for a very interesting talk. Our final speaker in this session is Ruth Schmidt, whose video I need to start.
Hello everyone, and welcome to my talk. Thank you so much to the organizers for giving me the opportunity to share some of my work here. My name is Ruth Schmidt, and I'm currently a postdoctoral fellow at the INRS, which stands for Institut National de la Recherche Scientifique, and a data analytics company partly based in Jojage or Montreal. My background is in microbial ecology, and my research largely focuses on tackling climate change issues in agriculture like drought by using microbes. And so since the beginning of this year, I've been working with Plotly on building DASH applications for the visualization of the data I'm dealing with that include amplicon sequencing data and all things omics. And this is what I would like to share with you today. So Plotly, besides being a company, is also the name of their open source graphing libraries that are available in R, Python, and JavaScript. Some of you might already be familiar with that or be using them already. And on your right, you see an overview of some of the interactive graphs that are available for the R open source graphing library. Dash is a very important product by Plotly that I'm gonna show you today and illustrate how a full Dash application looks like. So Dash is best described as a framework for building analytic web applications, which means that really anyone who wants to explore and visualize their data in an interactive way can use Dash to open their application in a web browser or host on a server to share with the community. Dash has been around for a while in Python, and since last year is also available in R. The great thing about Dash is that it's open source and MIT licensed, which means it's free. I put together a bunch of resources at the end of my presentation for you to check out if you want to learn how to build Dash applications yourself. And so the power of Dash for R is really that it combines R statistical models and graphics with interfaces that are highly customizable. I'm going to illustrate that later on in my demonstration. Core libraries include Dash core components that I'm going to explain in a second, and Dash HTML components that allow you to give a layout to your application. And there's also a few specialized libraries, including -bio for bioinformatics and -DAC, which stands for data acquisition and control, and includes things like a display for numbers that I'm showing also in my demonstrations later on. Dash components are kind of like Lego bricks that can be used to build customized apps and to interact with the app. And those core sets of components include things like a graph or a slider, a button, a drop-down, or even text in form of Markdown. And so in the next step, I would like to walk you through a few typical steps to build a Dash application. The first one, of course, includes loading the libraries. The second one is to start your app. And the third one defines the layout with some simple HTML. In the fourth step, you'd want to define your components that you want to see in your Dash application. So in this case, I defined a DCC input, which stands for text with a value that says hello, and I'm defining a DCC graph, which is a graph component to show a bar graph. And then this is where the fun starts in step 5, where you define the interactivity of your Dash applications in form of callbacks. And so in this callback, I am defining that whenever the value for the title changes, my figure output changes. In the second callback, I'm saying print me whatever I have typed in the text box. And then, of course, in order to see the output, I have to run the app on my local server in this case. And this is what the application looks like. So this is my text box, and I'm typing something in this case, hello bio 2020, let's dance. And whenever I type, my bar graph is being updated depending on the values I gave before. And as well, I can see the print here of what I typed in the text box. I can do all other things that I can do with a Plotly graph, including taking a snapshot, isolating traces, and zooming in, for example. So why Dash Bio? We're in a biology conference for developers. You all know that we're dealing with a huge volume of data, but also new and special formats of data. And so Dash Bio really aims to be a flexible and efficient UI builder for these kinds of data. It is fully compatible with the Dash4R package, and libraries that you're already familiar with maybe are Circos gQuery that we heard before, DSEC, another bioconductor package, and common packages that are used for microbiome analysis are available and can be directly connected with a Dash application and are in fact already used by many researchers and bioinformaticians. This is the Dash Bio Gallery, and you can head over and check out what kind of tools are already available, and these include, for example, a sequence alignment tool and a circular genome viewer based on Circus, heat map, and 3D models of molecules, for example. These are the resources I put together. Feel free to check them out. Slides are shared. And this wouldn't be possible, obviously, without a bunch of people, including Chris and Carson, who really laid the foundation for Dash R, and Ryan and Hamad, who were really like the main people behind the development of Dash R and the whole team of open source developers at Plotly, and Shamima, who really was the main person behind developing Dash Bio in Python and lent me some of her slides. Feel free to reach out to me as well if you're interested and have more questions. And now it's time for demonstrations. The first application I would like to demonstrate is an application available for statistical analysis and visualization of microbiome data using 16S amplicon sequencing data from my own research. And I followed here a typical workflow for the analysis of such data, including calculating alpha diversity and beta diversity to assess differences within or between samples. The packages I used for this analysis include packages like PhyloSeq, MicrobiomeSeq, iGraph for network analysis and visualization, and Picante for phylogenetic diversity. And then I plotted the output in form of interactive graphs. that you can explore by hovering over or isolating traces. If you were interested in a particular phylum that you want to see how it changes from control to treatment. And I can hover over the network graph as well and look at the connectivity of the nodes depending on date and treatment, and the same for the PCA plot. If I then wanted to interact with these graphs, I can choose different parameters from the drop-down menus. Here, for example, I can add another alpha diversity measure like inverse Simpson, and I can change from log scale to linear scale, and I can also explore how taxonomy or diversity changes over time. The same for plant soil compartments. In the second application, I would like to demonstrate a few -bio features. And this is an application I built for metabolomics data, in this case, volatile organic compounds, that I observe in my research a lot. And so a typical workflow consists of a principal coordinates analysis that can be visualized in a 3D space, or a volcano plot that you are all very familiar with for differential expression analysis that shows the magnitude of fold change or effect size here versus the log P. I can also use a heat map to visualize the values of a compound across all samples. And I can select specific compounds that I'm interested in from this drop-down menu. Then again, to interact with the graphs, I can select a date, and then all my graphs are being updated depending on the date I selected. Same for treatment. And for a volcano plot, I put a few extra settings where I can set my effect size to lower or higher, excuse me, or I can adjust the threshold. And depending on that, this is a very nice DAC feature that counts the volatile compounds, in this case, that are significantly up or down. So yeah, I hope I was able to demonstrate a few interesting features and show you what is possible with -R and -bio. Thank you.
Wonderful, very cool stuff. So just a reminder, keep the questions coming in the Pathable poll. And I want to take this opportunity to thank all 4 of the speakers for 4 very interesting talks. The first question for Anthony is, are there plans to expand the tool to other biological domains beyond cancer? For example, toxicological studies of environmental related compounds. Very interesting.
So we have plans to migrate toxicological data to the platform in the form of toxicosets, which are data objects that are generated from a package that we developed in our lab called ToxicoGX.
So the next question is for Shraddha. How is netDx different from SNF and MOFA, MixOmics, and is there a benchmark comparing these different approaches?
Sure. So SNF is also uses patient similarity networks, but it's an unsupervised method. It's used for integrating multimodal data and clustering the samples. netDx is for classification, so it's a supervised method. It needs labels. I saw a related question, is it only for cancer? But not necessarily. We've done asthma case control, we've done autism case control with rare CNVs. MixOmics, we have compared netDx to Diablo, which is in the part of the MixOmics suite. And the performance, so we've done a comparison of what pathways come up in performance, or what features come up in performance for these integrating tools. The performance was comparable for the benchmark we tested for netDx and MixOmics, but MixOmics tends to score individual features, like it'll say microRNA X is the top predictor, whereas netDx has that pathway capability. MOFA is also an unsupervised technique.
Great, thank you. Gabriel, how does pathway PCA compare to MOGSA or the PCA tools in Hadoga?
So MOGSA, is this, I wonder if this is Aideen's question. I think, so when I last compared pathway PCA to MOGSA, I saw really good behavior from MOGSA. So great work there. I didn't see that ours was horribly worse. So that's, I guess that's good news. To be honest with you, we haven't done a full suite of comparisons. We were sort of moving into the unmatched multi-omics meta-analysis world. And so a lot of these methods aren't used there. This result was some work that we did a couple of years ago. So when I saw that the MOGSA talk last year at BIOC 2019, I was pretty excited, but I was a little disappointed because I thought I was the only person in the space. So MOGSA is great. You should check it out. Also check out Pathway PCA. But the other one, which I saw in the chat, I had not heard of that one. What the see if I can scroll up. Oh, are the questions deleted after they're answered?
Yes, I think.
Oh, okay. What was the other one? Because I saw that one. I was in the middle of Googling it.
I think they are not deleted, but it's because there is a ranking because people can vote. So
okay,
they move up on the list. This might be the thing, but we have all questions.
Okay, well, I do not see, what was, if someone could tell me the name of the other.
Padoga?
Pagoda? Okay, all right. I have heard of it, but I haven't checked it out yet. So it is now on my list.
All right, the next question is for Ruth. Given Dash is from Plotly, does Dash offer different or better Plotly integration than Shiny?
Yeah, there's always a huge discussion between like Shiny and Plotly or Dash and what's the difference. Well, obviously because it is by Plotly, every Plotly graph can be integrated in the Dash application, but Plotly graphs can also be implemented in Shiny applications. We haven't so I can only say that it's a very easy integration with any Plotly graph or any ggplot that can be converted into a Plotly graph that can be directly linked to the callbacks.
So Anthony, does your tool support data with treatments by drug combinations?
So we are currently working on modifying the pharmacoset structure to better integrate drug combination data.
Great. Shraddha, what are the features in the network given that the nodes are patients?
The network is the feature. So if you have, for example, in my first example, you had the, clinical gene expression, some other omic, proteomic data and mutation data, and you make one network out of these, you have 4 features. If you take one gene expression matrix and break it up into pathway level features, you've got 2,000 features, and then feature selection will score each of them. So every network is a feature.
Gabriel, can you give a detailed explanation of the simulation data? You mentioned the outperformance of the global test is because it could well capture the association between biological and clinical features. Correct me if I'm wrong, but I'm not sure how the simulated data are close to real world data.
So that is a great question. We used the Pusher simulation study because it had just been published, and at the time it was the state of the science. After working through the Pusher data, we saw that it was lacking in some, I guess, biological certainty. So what we did then was the next simulation that we're working on right now is building straight from TCGA data. and we're using much more clinically relevant data to do this simulation. But at the time, we wanted to stick with what was published to show a reasonable apples to apples comparison. It's not. It's relatively simple multivariate normal data with some other multivariate distributions. So there's not an extreme level of biological sophistication to that simulated data for this simulation. The next simulation is, I've got one that we just finished running a couple of weeks ago, much more sophistication. So look forward to that in the next couple of months.
Ruth, can selections made in some panels be highlighted in others?
So if I understand the questions correctly, you mean if you select something in one graph, it would show up in another? But I assume that's the question.
I think so.
Definitely, you can connect different components with each other. So you can connect, for example, one graph component with another graph component. It all depends on how you define that in your callbacks, and you can have several inputs for one callback. So yeah, you could do that. It's very customizable. So whatever you wanted to connect with, you can.
Anthony, do you have any measure of data quality at the study level or cell line within study? How do you deal with data for ostensibly the same cell line in different studies? Is there any attempt to compare or combine through some sort of consensus these different studies?
So we currently do not have such measure. However, you can compare and examine the similarities and the differences of the drug sensitivity data and molecular profiles of the same cell line across the different pharmacosets using the pharmacogenomics bioconductor package.
Shraddha, do you envision applications also for single cell multi-omics data?
So there's nothing in netDx that makes it, specific to patient classification, because it takes a multi-assay experiment object as the data, and then it's, the sample metadata is in there. So if you wanted to, for example, classify cell labels, you could try it. And in fact, netDx does handle missing data. So you could try with imputation and without imputation of data. So, your nodes could be cell types, or they could be, whatever is relevant for other applications. Microbe classification, for example.
Wonderful. So we're at the end of our session. Please feel free to follow up with the speakers if we didn't get to your questions. All of the questions are in the poll. You can follow up through Slack or through Pathable. And thank you to all 4 speakers again for a very interesting session.
Thanks, Matt. Bye, everybody.